All right, shall we start?
Yes.
Okay. Well, hi everyone. Thank you for coming to the talk today. My name is David, and I'm a fifth-year PhD student from the Department of Biostatistics. And let me share my screen. Can you see the screen?
Yes.
All right. Okay. Thank you for giving me the opportunity to talk about the method and software FRAPOSA that I have developed. Is for a fast and robust ancestry prediction by using principal component analysis. So first, I would like to give a talk of the motivation of this method. The main goal for this method is to adjust for population stratification. So population stratification is a major confounder for genetic association analysis. And principal component analysis, or PCA, is a multivariate statistical method that finds the direction of maximal variability. So by aggregating information across genetic markers, PCA has been shown to be effective for population stratification adjustment. And we use the principal component scores, or PC scores, as an ancestry covariate to adjust for. And just a brief overview of principal component analysis. So in PCA, we find the direction at which the samples vary the most, at least the linear direction. And this can be computed by using a singular value decomposition. So suppose our data matrix is X, and it has P rows which correspond to the features, and N columns corresponding to the individual samples. And we also assume that X has been standardized, which means its mean is 0 and the standard deviation is 1. And we run a singular value decomposition on it to get the product U times D times B transpose. Here U is a P by N matrix, and it's our PC loading matrix. So each PC loading is a unit directional vector, and that tells us the direction at which the sample varies the most. And D is a diagonal matrix of the PC standard deviations, and we sort them in descending order, so the top PC corresponds to the most useful PCs. And the matrix V is actually what we are interested the most. That's the PC scores, the standardized PC scores. So by using that, we can adjust for the population stratification. And for computation, instead of doing a SVD, we can also use eigen decomposition if all we want is the U only or the V only. So that way we can speed up the speed a little bit. So commonly, I found that people use PCA by just doing a SVD on all the study samples they try to analyze. And here we use a alternative approach. We try to predict the study samples PC scores by using a reference panel. And this will allow us to integrate and match the study samples from different studies. And we can also use this method to predict the ancestry membership of each of the study samples if we have the detailed ancestry information in the reference panel. And there are several methods for PC score prediction by using a reference panel. The most straightforward one is we just call it simple projection. And then there is also a more sophisticated and also more robust method called, we call it augment, decompose, and procrastinate transformation. We call it the ADP. What we propose are two alternative methods, the bias adjusted projection and the online ADP. And we will start with a simple projection. So for simple projection, we first do an SVD on the reference panel. So this gives us the PC loading matrix U. And once we have the U, we know what is the direction that the reference samples vary the most. And we can simply project our new study sample onto the reference PC loadings. And since the PC loading is already a unit vector, we can simply multiply the new sample x new to the PC loading matrix uref. The biggest advantage is that it's computationally very fast. If you have M samples, then and you want the top K PCs, and you have P features, which usually corresponds to the minor allele count of the SNPs, then the whole computation complexity is just O of M times K times P. And also, this method is very simple to implement. But the downside is that the study PC scores will tend to shrink towards 0 when the number of features greatly exceed the number of samples. So essentially, this is an overfitting issue. Once you have too many features, once you have too many genetic markers in your reference samples, instead of looking for the maximum direction of variability for the true signal, the PCA actually starts to explain the noise in the reference panel. Of course, this is not transferable to your study set. So as a result, the PC loadings for the reference panel actually cannot separate the study samples very well. So as a result, you can see that the study samples PC score will be much closer to 0 than in the reference panel's PC score. And I will show you examples later. And there are many softwares that implement this method. You can use plain Eigensoft or just write your own code. You can probably get it done in 10 or 20 lines of code. And a more robust method is the ADP method. So what we do here is quite similar before in the earlier stage. So we first find the reference covariance matrix, xref transpose xref. And then we do an eigen decomposition on the reference covariance matrix. And this gives us the reference PC scores. And then every time we have a new study sample, we will just append this new study sample to the reference panel. So now the reference panel is has one extra sample. And then we just find this the covariance matrix of this augmented matrix. And then we do a eigen decomposition on the covariance matrix. So now you have a PC score matrix VAUG. So this VAUG and the VREF, the dimension is different only by one for the new study sample. And as you can imagine, since you only have one more sample, the rest of the matrix of VAUG actually is very close to VREF. And if we take only the last row of this VAUG, then that is just a PC score of the new study sample, and that's what we want. So essentially, we have a reference sample that's fixed, and every time we have a new sample, we just add this sample to the pool and do a PCA, and that way is the new sample PC score is always calculated based on the reference samples. And after that, we find a linear transformation that minimizes the difference between f of VAUG ref and the VREF. So VAUG ref is simply the top n rows in VAUG. So that corresponds to the reference samples. Because we added a new study sample, so the PC score changed a little bit. So we just want to restore this slight difference to the original PC space. And once we find this transformation, this linear transformation, which is called a Procrustes transformation, we just apply this transformation to the PC score of the new sample, and then get the PC scores in the original PC space. So that is how the ADP method works. And the One of the greatest advantage of the ADP method is that it has a higher accuracy and has a minimum shrinkage, different from the simple progression method. But the downside is also very clear. It is very computationally costly for a large reference size. So because every time we need to do every time we do the eigen decomposition on the augmented matrix, we are essentially running a n-cubed algorithm we are essentially running an n-cubed algorithm. So the total computation complexity is m times np plus n cubed. M is the number of your study sample. So for each study sample, the complexity is the reference sample size cubed. And when you have lots of reference samples, this is very slow. But oftentimes when we need very fine-scaled ancestry information, we do need a very large reference sample. So ADP has a big computation limiting factor here. The software that implements this method is the trace software by Chaolong Wang, who used to be in the biostats department at UFM. And any questions so far? Okay, I'll continue.
Yes. So, what is ancestor prediction, and how is that related to PCA?
So ancestry prediction, essentially, we want to know the ancestry information of our study samples because that can be a big confounder in the analysis. So one way to record ancestry information is simply do it categorically. So say this sample belongs to European, that sample belongs to the African, and but what we do here is actually
before you go on, I'm sorry, but what does ancestry sample mean? And actually, what is the biological problem you want to address? Not exactly clear.
For example, in GWAS analysis, we always need to adjust for the covariates, and ancestry information is quite often is a confounder. So the effect in the Africans is quite different from the genetic markers effects on the Europeans, say. And so we need to adjust for this the difference across different ancestry groups, and that's the main motivation.
So here, when you say ancestry information, it sounds more like the difference between different cohort rather than rather than predicting its ancestor. So
yeah, you can call it population stratification. So there are different population groups, and they have they will make a difference on the effects in for the variants you are the most interested in.
okay, so we need to adjust for the ancestry information. Then why do you call it an ancestry prediction problem? Because when you do GWAS, you always know what is your cohort, well, whether it's African or American or Asian.
If you do know your participants' ancestry information, then that's good. And yeah, that you can just use the categorical data directly. But there are cases when you don't know that information, and all you have is genotype samples. In that case, we can just use the genotype samples to predict the ancestry information. And there are also cases that you have some vague idea, you have some vague information about the ancestry, like you know this person is European or non-European, but nothing else. But you would like to get a more detailed information about the non-European groups, whether they are African or Asian. Or maybe you want to know better about within your European group, whether they are British or Finnish or Spanish. And in that case, if your reference panel has detailed ancestry information, you can use that to predict the ancestry information of your study samples. And also there are cases when the self-reported ancestry information in your study sample is not is questionable. So in that case, you want to use the genotypes instead. And also, if people don't want to use the categorical result, but use numerical covariates to adjust for instead, then PCA will also be useful.
Okay.
Yeah, thanks for the question. Any other question? Okay, I will move on. So after overviewing the existing methods, now I'm about to address our proposed methods. So one of our proposed method is we've called the bias adjusted projection. So that, remember, in the simple projection method, we have a shrinkage effect going on. And a simple way to adjust for that is to estimate this shrinkage factor and then pull back all the PC scores. And another approach is to improve the ADP method. So ADP method is quite accurate but slow. So we just try to improve its speed. Remember, every time we do an eigen decomposition on the augmented matrix, all except the last entry are the same. So there is a lot of repetitive computation going on. And if we do some smart linear algebra, we can greatly reduce the computation time. And we implement both methods in the FRAPOSA software, which is available on GitHub. And now more details about the bias adjusted projection. So we first do an SVD on the reference samples. And after we do the SVD, we would estimate the magnitude of this shrinkage for each PC. We call it gamma 1 to gamma k, where k is the number of PCs you want. And we use the asymptotic properties of the generalized spiked population model. And by using this method, we can compute, we can estimate the degree of shrinkage by using the eigenvalues. Essentially, we are assuming we are assuming a distribution for the matrix, and then we will try to estimate the degree of shrinkage and angle of shrinkage under this assumed distribution. And after we find the shrinkage factor, we will still project a new study sample to the reference PC loadings. But after doing this projection, we will just adjust for the shrinkage and get a new PC score. So we can just doing a multiply the old PC scores by a diagonal matrix of the inverse of the shrinkage vector, and that will pull back all the PC scores. The computation complexity is the same as simple projection, which is the O of m times k times p, so it's very fast. And also notice that when we calculate the shrinkage factor, this shrinkage factor is particular to the reference set only. So it is a computation that's done once for all. So it doesn't affect most of your computation time. And the software that does this is, there is an R package called HDPCA, and we also wrote a Python wrapper in the FRAPOSA software. And the next proposed method is the online augment, decompose, and processes method. So in this method, we also do a SVD on the reference matrix first. And then every time we have a new person coming in, we will use the online SVD algorithm to update the original reference PC score matrix to the new PC score matrix. And for doing that, we need the PC loadings and the eigenvalues of the reference matrix, and also of course the new samples. And I will show the details of the algorithm in the later slide. And this method was originally developed by a researcher in image processing, but we found that method is quite useful in our case about genetics data. And after we do that, it's the same as the ADP before. We would split the matrix into the samples that correspond to the reference samples and the part that responds to the new samples. And again, finding a procrustes transformation to project the PC scores of the new sample. And yeah, that's how the OADP method works. And what we found empirically is that the outputted PC scores are very close to ADPs. But the computation is much more efficient. Although ADP had a complexity of O of m times np plus n cubed, in our case, the complexity is only linearly with respect to n, which is our reference size. So the OADP is much faster than ADP when the reference sample has when the reference size is very large. Essentially, the speed up comes from the almost repetitive eigendecomposition of the n+1 by n+1 augmented matrix for each study sample. And it also avoids computing the bottom PCs, because in a standard SVD, we decompose the whole matrix. That will give you all the PCs, but you usually only need the top few ones to use to adjust for ancestry. We also find that this method is very effective when approximating the top PC score from ADP. And for example, if you need top 4 PCs, you can just compute the top 8 PCs by using the online SVD method, and that will even make the accuracy higher. If you do compute all the PCs in the online SVD method, then the speed of OADP is identical to ADP's. So don't do that, and you also don't need that. And our software FRAPOSA implements this method. And here is a more detailed description of the online SVD method. Essentially, it is a bunch of quite clever linear algebra on your reference matrix so that you can update the old reference matrix to the augmented one without doing much computation. And I won't go too much into the details here, but the slides will be available later on, and you can look into for more details if you're interested in. So here is a table to summarize the computation complexity of each method. You can break down the runtime into a reference part and a study part. For decomposing the reference matrix, all the methods have the same complexity because we always need a SVD on the reference panel to use PCA. But the study complexity is very different. You can see that for SP and AP, the complexity does not depend on the reference sample at all, which is n here. So it's each method always the complexity is always linearly proportional to your study samples, which makes sense. So each study samples has the same runtime. And for SP and AP, the more pieces you want, the longer it takes, and also the more markers you have, the longer it takes. But for ADP, it's quite slow. So if the reference size is larger, then your study complexity will goes up cubically. But for OADP, this would only go up linearly, which is much more manageable. So here we have some simulation studies to demonstrate the performance of each of the methods. we use a coalescence-based grid simulation approach with population migration. We simulate 4 populations on a 2x2 grid. We first generated the haploids and then paired them up into diploids. And the migration rate here is set to quite large so that we can test the fine scale population differentiation. There are 200 samples are randomly selected as the study samples, and the rest are for the reference. And the reference size, we changed it from 1,000 to 3,000 to see how the reference size affects the accuracy and the efficiency of each of the algorithm. We compute only the top 2 PCs since it's only a 2x2 grid. And to measure the accuracy, we scale the mean square difference between the reference and the study population centers. So if the reference population center is close to the reference, if the reference population center is close to the study population center, then that's good. That means your study samples is not drifted away far from your reference samples. But if it's far away, especially if it's string towards 0, then that means it's the reference samples, the reference PC is not good at explaining the variation in the study samples. So here is the result. In each of the 4 panels, one method is shown: SP, AP, OADP, and ADP. So the colored circles are the colored markers are the reference samples. So we have 4 populations here, and each of the reference sample from each population is colored differently. So you can see we have the green reference sample, the red reference sample, the cyan reference sample, and the blue reference sample. And I also draw a circle draw a circle that covers 95% of the reference samples to better visualize the center of the population. And the black markers are the study samples. These are the samples with unknown ancestry, and we use PCA to predict their population stratification. So you can see that in the case of SP, the study samples has visibly drifted away from the center of the population, from the reference population towards 0. But on the other hand, if we look at the AP and OADP and ADP, you can see that the reference samples and the study samples are much closer to each other, at least within each population. So this is the kind of the way we want things to be. And for SP, when things are drifted towards 0, it's just things are less accurate. So in this case, although you can visibly see the shrinkage effect, the damage is not that big because each of the study samples are still far away from each other. But you can imagine if the number of markers go larger, then these 4 black circles will be very closely clustered, and that's what we cause a lot of mistake in ancestry information prediction. So this is a scatter plot of each of the 4 methods, each 4 methods PCs, and also we have there the mean square distance between the PC score of each method. So you can see that SP, as we had expected, has shrinked its PC score closer to 0, and SP is also PC score also far away from the other 3 methods, and the other 3 methods have almost identical PC scores, and their square difference is also very short. And here is I'll show you the effect of the reference size on the PC score prediction. So this is the plot for using a reference panel of 1,000 samples. And if we increase the reference size to 2,000, you can see that the shrinkage effect for SP is less severe than before. Before it's much closer to 0 and far away from the reference center than now. And for the other 3 methods, the change is not that big different. And this is consistent with what we had said earlier. The shrinkage effect of simple projection is the most severe when the number of features greatly exceeds the number of reference samples. So when the reference size increases, the shrinkage effect also gets milder. And when it increases to 3,000, SP's performance is not much lower than the other 3 methods. So this is the plot of the for the accuracy of each of the 4 methods. And the y-axis is a MSD, the lower the better, and the x-axis is the reference size. So you can see that as we change the reference size from 1,000 to 3,000, the for AP and OADP and ADP, the performance is about the same. But for SP, it really, you can see that it's quite sensitive to the reference size. And this plot is the axis is the same as before, but here we show the runtime, the empirical runtime of each of the 4 methods. And this time the ADP is one that singles out from the other 3. You can see the runtime increases much faster than linearly compared to the other 3 methods, which look almost flat in this plot. And this is a table this is a table. It's the same numbers for that generated previous plot. So you can see that SP and AP is very fast. It took only a split of second to get the results for each sample. And for OADP it takes a longer time, but not that slow. But for ADP it's much slower. We then applied our methods to the UK Biobank data to see how it runs on the real data. And for our study data, we have about 148 variants for 488 individuals. These samples are collected from multiple centers in the UK. For the reference panel, we used the 1000 Genomes project. We have 84.4 million variants, and that covers most of the variants in the UK Biobank. We have 2,492 individuals where the children have been removed since they are related to the parents. There are 26 populations, which all fall into the 5 superpopulations, which are Africans, Americans, East Asians, Europeans, and the South Asians. Then we do an ancestry membership prediction. We use the top 4 PCs, and we used a majority vote among a study sample's nearest 20 reference samples. So essentially it's the K nearest neighbor method. And also we are interested in, later on we are interested in finding the finer scale ancestry information inside each superpopulation. And so in this case, we determine whether a study samples has a mixed ancestry background. So if the study sample's highest vote, the percentage is less than 7/8, so then we classify it as mixed. And then we run the PCA on each predicted population separately to obtain the finer scale ancestry membership. So this table shows all the populations and the number of samples available in the 1000 genomes project. So you can see the number of samples are more or less evenly distributed among the 5 superpopulations, and inside each population, we can get we also have the smaller subpopulations. So this is the PC scores for the reference samples, for the 1000 genome samples. So the left panel is the PC1 and PC2, and the right panel is PC3 and PC4. And we colored each superpopulation differently. So you can see that the European is a cyan, so it's here, and the Africans are the blues as the left bottom corner, and the Asians are on the, oh sorry, I said it's the other way around. The Asians are at the bottom left, and the Africans are on the right. And here in the middle, you have green, which is the admixed Americans, and also the South Asians, which is purple. So you can see that PC1, PC2 already separate the African, Asian, and Europeans quite well. And if you look also into PC3, you can see that PC3 mostly separates the Americans and the South Asians from the other 3 groups. So we can so you can see all the 5 populations, all the 5 superpopulations are very well separated in the top 4 or just top 3 PCs. And we also computed the shrinkage factors for each of the top 4 PCs, and it's very minimal. Even PC4 only have a 0.94 shrinkage. And now we project the study samples PC scores on the reference PC space. So yeah, now we have much more samples, and we also add a group of samples of mixed ancestry information. So these colors represent the predicted population of the study samples. And the reference samples now are color gray in the back. So these 2 panels are done by SP, and we compare that to APs. So you can see that there is some shrinkage that has been adjusted. But in this case, the shrinkage is not as strong, so you can't see the difference that clearly. Later on, we will see how what caused the shrinkage worse. And if you compare that to OADP, it's pretty much the same. These 3 methods are the same. And we didn't include ADP here because ADP takes a lot of time to run. And in our small sample study of the UK Biobank, they have almost identical results. So OADP and ADP are both look like this. So this is the table that summarizes the number of UK Biobank samples that is predicted to belong to each of the 5 superpopulation. And as what we can expect is most of them are predicted to be Europeans, and also there are also a lot of predicted to be South Asians and Africans. And now we look into the results inside each superpopulation. So this is reference samples for the Europeans. These are the 1000 genome samples that are labeled as of European ancestry. So inside European, we have 5 populations. So these 2, the red and the blue, are the blue is British, red is 1000 genomes, call it the Utah residents of Northern and Western European ancestry, so which is pretty much British. So these two groups are almost have the same ancestry membership. And here on the right is a fins, so it's quite far away from the others. And down here, the cyan and the purple are the Spanish and the Italian, respectively. We also included the PC3 and 4 here, although they're not that useful in this case, although PC3 can probably separate the Spanish and the Italian better than just using PC1, 2. So this is the study PC scores and their predicted ancestry membership that's done by SP. And you can see that the reference samples are colored in gray in the back. So the study samples which are colored with different colors are clearly shrinked away from the reference ones. If you look at the results by AP, now the difference is pretty big. So in the case of SP, you can see that many of the studies, since they're all shrink toward the center, they're classified as British or Utah of Eastern and Western and Northern European ancestry. So this will cause a lot of misclassification. Now compared to OADP, AP and OADP are very close. You can see that. If you look at PC3 and 4, OADP actually separates the study samples slightly better than AP. But these two are, both are much better than just using SP. Yeah, something like here, PC3 and PC4 is all clustered together. That's something you really don't want for adjusting for population stratification. And this is a summary of the accuracy of each of the 4 methods. So the reference size, so we in the first column is a study of all the global samples. So in the global samples, we have about 2,500 reference samples from each of the 5 superpopulation. And we randomly selected 5,000 study samples. And then we do a PCA on the reference and the study samples, and we find their shrinkage factors. And here, and also the FST, the fixation index, that shows the population differentiation inside each population. We also include the PC1 to 4th contribution to see how much it can be explained. The variation can be explained by the top 4 PCs. And yeah, in comparison, you can see that since the European study, inside the European study, we only have about 500 reference samples compared to the global study of 2,500 samples. The number of reference samples is smaller, and also the difference between the population differentiation is also smaller, as you can see by the fixation index. So because of that, the shrinkage factor become much stronger in the European samples than in the global samples. And you can see the top European, the PC1's shrinkage factor for the European samples is already as bad as 0.7 when the global sample is only 0.99. And for PC4, the shrinkage is 0.14. So that's like 1/7 of the original reference scale. So that is very bad. And also the top 4 PCs contributed less than in the global case. And you can see that as a result, SP's performance in the European sample analysis is much worse than in the global sample analysis. On the other hand, the other 3 methods have about the same performance. And this table shows the runtimes for the for all the UK Biobank samples. So we run SP, AP, and OADP on all the UK Biobank samples, but for ADP, we only run it on 5,000 randomly selected UK Biobank samples, since it will take a very long time to really run it. But we just linearly scale the runtime to predict how long it will take if you really run that many samples. So if you first look at the global analysis, SP and AP took less than 1 hour to finish almost 500,000 samples, and OADP took slightly more than 20 hours, which is also doable. It's just overnight. But ADP is if you really want to run it, it will take you 1,600 hours, which is not very practical. And for the Europeans, it's because the reference size is only 500. OADP and also OADP has a smaller runtime, and ADP's runtime is greatly reduced. In comparison, SP and AP is the speed is about the same compared to the global analysis, since it doesn't need its computation complexity does not depend on the reference size. So to wrap up the talk today, so we compared 2 existing methods, SP and AP, and 2 novel methods, AP and OADP, for predicting population structure by using PCA. The OADP and AP are much faster than ADP when the reference size is large. For OADP, the complexity of OADP is O of n, and for AP is O of 1, but for ADP is O of n cubed with respect to the reference size. In the UK Biobank analysis, the runtime of OADP is 80 times faster than ADP, and AP is 2,000 times faster than ADP. And for accuracy, OADP and AP do not have the shrinkage bias, unlike SP. And SP shrinks many European samples towards 0 and cannot distinguish them, while OADP and AP have successfully separated the European samples at finer levels. The software for all the 4 methods and the tutorials are included on the GitHub for FRAPOSA. It's written in Python, and you're free to, I'll be happy if you find it useful and then play with it and give me some feedback. And we also have a paper here for the method and the data analysis I have talked so far for your reference. And that's all. Thank you. Any question? Questions, comments, anything you are curious?
Oh, thank you, David, for a good talk. We appreciate you starting off our fall semester Tools and Technology seminar series.
All right, thank you for organizing this. All right, then I guess this is the end of the talk. And if you have any question, you feel free to shoot me an email.